Hi everyone. This is the second mini lecture in Chapter 7 on microbial metabolism. We're going to be talking about microbial energy today. So we'll be looking at how cells pursue energy and then how they utilize that energy once they have it. Cells require both a constant input of energy and they also require a constant output or expenditure of that usable energy. Energy can come in the form of light for those microbes that can perform photosynthesis, or it can be contained in chemical bonds that will be released when those substances are broken down by the cell. Generally speaking, energy is stored in the molecule ATP, or adenosine triphosphate, and only chemical energy can routinely drive cell transactions. So it's how within a cell, that's the form of energy that is used for anything going forward. Chemical reactions are the universal basis of cellular energetics. So it's those chemical reactions that actually drive everything in a cell. So energy in cells is managed in the form of chemical reactions, and that can involve the making and or the breaking of bonds and then the transfer of electrons. So we can break that energy down into those reactions down into two different categories. The first is called exergonic. Exergonic reactions actually release energy and make it available for cellular work. So this is what we would normally think of breaking down perhaps a glucose molecule. It would make that energy then available for the cell to get some work out of it. The second form of reactions we see in cells are called endergonic reactions, and these are driven forward only by the addition of energy. Generally speaking, that's in the form of ATP. So these two types of reactions are often coupled together, and that is so that when you have an exergonic reaction and you've released energy, it's readily available to be used then in an endergonic reaction that requires that energy. So cells extract that chemical energy that is present in that nutrient that they have somehow managed to get inside of them, and they apply that energy towards useful work. That may be building macromolecules, that may be some sort of function that the cell must do, and it needs energy to do that. Cells possess enzyme systems that will actually trap that energy in those bonds of nutrients as they're broken so that it can be used then or stored for when it's needed later. So during those exergonic reactions, when that energy is released, that energy then will be stored in the high energy phosphate bonds, especially the molecule ATP. ATP is what fuels those cell reactions that need energy. So I want to talk to you a little bit about oxidation and reduction. Hopefully this is review for most of you. If not, this is something you might want to spend a little bit of time looking through the book. Oxidation and reduction are types of reactions. Oxidation would refer to when there is a loss of electrons from a compound. When it loses those electrons, it is said to be oxidized. A good way to remember this when we're moving on to reduction. Reduction is the gain of electrons. So when a compound gains electrons, it's considered reduced. I always remember that as gaining electrons. To me, that means more negative, which means reduced, getting smaller. So oxidation reduction reactions, often referred to as redox reactions, are very common in the cell and are one of the most important ways that we have for required energy transformations. So it's a way for the cell to break down bonds, but then carry those over to where that energy is needed from the energy released from breaking bonds. So we have these enzymes called Oxido Reductases. These are enzymes that will remove electrons from one substrate and add them to another. Often what we see in the cell are two. One is called NAD, the other is FAD, or nicotinamide adenine dinucleotide or flavin adenine dinucleotide. These are two that are very good at being able to accept electrons and then give them up. So they're kind of a shuttle. And in a redox reaction, we will have a redox pair. That means that one of the molecules is going to be an electron donor, and then you will also have an electron acceptor involved in that. And this is a great example of that, kind of a very simplified form. Hopefully for those of you who've had chemistry, this will be a little bit of a review. When we look at sodium chloride, if we look at the sodium atom on its own, it has one electron in its valence shell up here. This is not very stable, and the sodium is not happy with this. It wants to fill its outer shell, either get rid of this electron, or you would want to add a bunch. So if it is mixed in with chloride, which has 7 in its valence shell, the outer shell, it also wants to have 8 there. What these two atoms will do when they're together is the sodium will give up an electron, and it will become oxidized because it's given up an electron. And the chloride will accept that electron, filling its outer shell to 8, and it will become reduced. So you often see this where one atom will give up to the other, so one will be the oxidized and one will be the reduced. This can also happen, though, in more complex molecules with the same idea, that it's giving an electron and the other one is accepting it. So when we look at oxidation and reduction, there's energy present in that electron acceptor that can be captured. So when it accepts that electron, there's actually energy there, and it will allow it to phosphorylate, which means to add an inorganic phosphate to ADP. So we have adenosine diphosphate here, and we want to add that inorganic phosphate, which would give us then ATP, which is adenosine triphosphate, and that's a great way to store energy in the cell. The cell doesn't really handle electrons by themselves. They're really part of an atom. So they're often considered a hydrogen atom, which consists of a single electron and a single proton. But really it's the electrons we look at for transferring that energy. And then at the end of a redox reaction, or in a redox reaction, we can have dehydrogenase. That is the removal of hydrogen during a redox reaction. So taking a hydrogen and removing it. When we look here, we have some electron carriers that would be commonly found in a microbe. These molecular shuttles actually can go from loaded with an electron to unloaded, and so they can repeatedly accept and release electrons and also hydrogens to facilitate transfer of this redox energy. So it can start out with this NAD+. We then add 2 hydrogens, which are really 2 electrons, and this will give us this energized molecule here that then can be transferred to an ATP. So it's a great way to kind of transfer energy from a molecule to another one. And that brings us to ATP, or as the book likes to refer to it, metabolic money. This is kind of where we get that energy to be saved and then to be spent at a later time. Adenosine triphosphate is a 3-part molecule. We'll start over here. This is the nitrogen base, which is called adenine. It is connected to a 5-carbon sugar here, which is ribose. So we have our adenosine and we have our ribose here. So when we add them together, our adenine and our ribose, which when you add them together is adenosine. And then off of the ribose, we're going to have 1, 2, and 3 phosphates. These phosphate groups are bonded to the ribose, and it's actually the connection here between the 2nd and 3rd phosphate and here between the 1st and 2nd. These are very high energy bonds that when they're broken, they're going to release energy. So the removal of that terminal phosphate, if we go from adenosine triphosphate to adenosine diphosphate, will release energy. And this is how then that the cell will have energy to make other reactions go forward that need energy. So when we're talking about the metabolic role of ATP, ATP is utilized and replenished in an ongoing cycle, meaning that we're using it up when we need energy, but then we're quickly replenishing it when we're breaking down nutrients and we're storing it for later. So energy released during the ATP hydrolysis powers this biosynthesis, what allows the cell to make macromolecules and to do its functions. When ATP is utilized or used, that terminal phosphate is removed. I talked about that. That terminal phosphate getting removed releases a lot of energy, and then you have an ADP that is left. You will need to put in energy to replenish that ATP, and that may be from the breakdown of some sort of cellular fuel. And in heterotrophs, the catabolic pathways, so the pathways that break down nutrients or macromolecules, provide that energy infusion that will generate that high energy phosphate. So it is what will take the ADP and phosphorylate it to make it ATP. So it's this ongoing cycle of breaking down ATP to release energy and run reactions that need energy, and then it's the breakdown of those nutrients coming in that release energy and allow us to form ATP for later. So this is a brief introduction into redox reactions and the role of ATP. I'll go into some other reactions in the next mini lecture.